Welcome back. This is part 2 of mitosis and meiosis. We're going to talk about meiosis or gamete formation. You should have already learned about mitosis to have a little bit of background for this. Now, the things that you need to know are the role of meiosis and fertilization in sexually reproducing organisms, the importance of homologous chromosomes to meiosis, how the chromosome number is reduced from diploid to haploid through stages of meiosis, the 3 important differences between mitosis and meiosis, and the importance of crossing over independent assortment and random fertilization to increase genetic variability. You'll notice that there's an awful lot of vocabulary terms in there. Make sure you learn your vocabulary for this unit. It's going to be very important. A gene. Gene is just a segment of DNA that codes for basic units of heredity. So on our DNA, we have little pieces, and those pieces each code for something. It could be a code for your eye color, a code for your blood type, all different things in there, but each little one we call a gene. Offspring acquire genes from parents by inheriting chromosomes during reproduction. Now, offspring, that means babies. So our babies get genes from their parents as we reproduce. There are 2 different types of reproduction. We have asexual and sexual. Now, asexual reproduction is one parent, just one, and it's creating a genetic identical copy. We can also call those clones. Now, there's very little variation because we're making a copy. Very little variation, only a little bit, and that's due to mutation. We're going to talk about that more in their next unit. This type of reproduction is pretty quick, and it's very energy efficient. And examples of this include budding, binary fission, and then of course there's mitosis, which we talked about in the last video. Asexual reproduction takes 2 parents, not just one, you have to have 2. And we have a male and a female that produce gametes. And when those gametes come together, we form a new individual. This produces a lot of diversity and variation in populations, but it's slower and much more energy consumptive. It takes a lot more time and a lot more energy to make those babies. Now, asexual reproduction can take place in as little as 20 minutes for bacteria. But if you think about like people with sexual reproduction, that's 9, almost 10 months. And think of elephants. Elephants are 2 years. It takes a lot more time and a lot more energy going in to build whatever that baby is. Now here we have some examples of both asexual and sexual reproduction. First here, we can see that we have a bacteria that is undergoing binary fission. It basically just copies the DNA, moves the 2 copies to the sides, and then cuts in half. This one here, this is budding. This is called a hydra. It lives in aquatic environments. Now this hydra is photosynthetic, and you can see here that it's growing, and then off the side of it, we have what we call a bud. And this bud is going to be just more cells like this. It's going to produce, and then it'll fall onto the floor of the environment, and a new one grows, just like its parent. Now here we see redwood trees. Now we tend to think of trees as being pretty similar. We'd look at all these redwoods and think they all look pretty much alike. But in fact, there's a lot of genetic diversity. These reproduce sexually. And then we have here our elephants. And as we talked about, they do reproduce sexually. It takes a lot more time to make a baby elephant than it does to make a hydra. Now we need to talk about chromosomes. We have 2 different types of cells in our body. We have what are called somatic cells or body cells, and these are diploid or what we call 2N. Okay, so diploid and 2N mean the same thing. And for humans, our diploid number is 46. We have 46 chromosomes. Now 2N, that refers to the number of copies. We get 1 whole copy from mom of everything and 1 whole copy from dad of everything. And together, that's 2N. So we got 23 from mom, 23 from dad, gives me 46. Each pair we call homologous chromosomes. We get 1 again from mom, 1 from dad. And they go together. So they're homologous, but they're not identical. It's kind of like shoes. You have a left and a right shoe, and they go together, but they're not identical. Now, Autosomes are chromosomes that code for everything except for the sex, okay, male or female. So we have 22 of those in humans. Now, this is different for other species, but in humans, we have 22 autosomes. And then we have one pair of sex chromosomes. These are X's or Y's. Again, for humans, other creatures have different systems. And in humans, females are XX and males are XY. Now, we have recently learned that there's a lot of variation that can be occurring here in our genders for XXXY and how we determine sex at birth. Now, for gametes, that's the other type of cell that we have in our body. And these are our sex cells. So these include eggs and sperm. These are what we call haploid. They have half the information of a somatic cell. So they're N, haploid. And since we have 46 chromosomes in a regular somatic cell, gametes have 23. 22 are autosomes and one sex chromosome. Now you'll see up here, we have 22 pairs of chromosomes, and this says that we have 22. That's because we get one of each. So if we get homologous pairs, one from mom and one from dad, our gametes only get one. They don't get both. And then they have a sex chromosome. Now an egg in humans is 22 autosomes plus an X because females only have an X to give. If we look at sperm are 22 autosomes plus an X or a Y because males have an X or a Y to give. Now here's looking at homologous chromosomes in a somatic cell. So this is just a body cell. And you can see here that this organism is 2n equals 6. That means its diploid number is 6. It has a total of 6 chromosomes. If I know the total is 6, that means it has 3 pairs, 3 from mom and 3 from dad. So n equals 3. And here you can see that we have one really big one, one medium one, and one small one that are blue, and the same for red. Now, if we look here, we have the chromosome with its sister chromatid. So this is a chromosome that's already been copied or duplicated. So it's there, and then it's got a copy with it. That's why we see 2 lines. But this one has one that matches in red. These are the homologous pair. Oh, and then here's another homologous pair. And we have our little ones here. These are homologous pair. You may have heard of a karyotype. A karyotype is an image that's made of the DNA chromosomes, okay, within an organism's cell. So we find the cell in mitosis, okay, undergoing replication, where all the chromosomes have condensed into that packaged look to them. And we then take pictures and we arrange it from largest to smallest and match them up. Now you'll see here that this individual has 22 plus this here. We have an X and a Y. Because it's an X and a Y, that means it's a boy. If it was an X and an X, that would mean a girl. So this represents the DNA of one individual. You've probably heard of this being done for babies inside mom. They'll do an amniocentesis where they take some fluid around the baby and then they find cells in that, and they can do the karyotype. Now we do this not just for babies to see if they're okay, but here's a karyotype of a cancer cell. Now this comes from a metastatic melanoma. Now that means that this is a skin cancer that has metastasized or it's spread through the body. And you can see here that there's a lot of strange things going on. There's 3 number 1s, a whole bunch of number 2s. Look at this number 7. Okay, weird things going on. But if you remember back to cell signaling, we know that cancer is a problem of the cell cycle and of the division within the cell. Okay, so when mitosis goes wrong, we can get weird things like this. The life cycle for an organism is from conception to being able to reproduce its own offspring. That's the life cycle. So fertilization and meiosis alternate in a sexual life cycle. So for humans, we have fertilization and then meiosis. Meiosis are cell divisions that reduce the number of chromosomes from diploid to haploid to create our gametes, eggs and sperm. Fertilization is when we combine gametes, so sperm from one organism and eggs from another. And when we put those together, we come back to diploid, 2N, and we call that a zygote. Now, believe it or not, there are organisms that are sexual that the sperm and egg come from the same organism. Lots of plants do that, where they actually fertilize themselves. A zygote then goes on to divide by mitosis to make a multicellular diploid organism. So here's our life cycle in a diploid multicellular organism. Here we have a mouse. So this mouse is going about its 2N life. Then we have meiosis occur, and this creates our gametes, eggs or sperm. It combines with another gamete for fertilization, and then mitosis takes place. Then it goes back to meiosis, fertilization, mitosis. Here we have a short video here from the Amoeba Sisters. These are a great set of videos. I highly recommend watching them. Okay, so this one is on meiosis. After we watch this video, then I'll talk about the actual stages of meiosis.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible.
So those are the different stages of meiosis. I hope that kind of made sense. We're going to go through it together to hopefully clear up any misconceptions or misunderstandings. So meiosis is reduction, division. We need to divide and reduce the amount of DNA that's present. We wind up with 4 daughter cells, but we have to divide twice to make that happen. So here we see meiosis 1 and meiosis 2. Before any of this happens, we have interphase. During interphase, our DNA is replicated. We wind up with our duplicate. So we have our original piece of DNA for all the different chromosomes, and it makes a copy, and they're going to hang out together. Now for meiosis 1, during prophase, we have synapsis that occurs. This is where the homologous pairs hang out together in a tetrad. Tetra means 4. If you've ever played the game, there's 4 blocks in each one of those different sheets. So we have our 4 together in synapsis. And because there's so much here, they can kind of tangle around each other and then have what's called crossing over, where they swap pieces. Okay, and that happens in prophase 1. Now, during metaphase 1, they're going to line up in their pairs. So we keep the buddy system. But then during anaphase 1, those buddies are split up. Now notice they still keep their copies, but they split up. Then we wind up making 2 cells. So now we need to divide again, but we don't do interphase again. No interphase, because during interphase, we make copies of the DNA. We're trying to get less, not make copies and have more. So we go from telophase 1 directly into prophase 2. Now, prophase 2, metaphase 2, anaphase 2, telophase 2, all of those are just like mitosis. You already learned it, so you don't have to learn more. So, prophase 1, we have synapsis and crossing over. Metaphase 1, they lined up with their buddies. Anaphase 1, they pull apart. Telophase 1, we make 2 nuclei. Then we have prophase 2. And in prophase 2, they pack back up, make sure they're with their copy, and then they line up single file for metaphase 2. They separate from their copy in anaphase 2, and now we make new cells. So that will give us 4 different cells. Now, the things that are unique in meiosis, so we see them all in meiosis 1, is that during prophase, we have that synapsis and we have crossing over. We don't see that in mitosis. During metaphase 1, they line up in pairs, not single file. And then in anaphase 1, we separate the homologous pairs, but the sister chromatids stay attached to each other. Those copies are still together. Then we go to telophase 1 and then into meiosis 2, which is just like mitosis. Okay, here's our quick way to kind of look at those is we have interphase always happens first, then we have prophase 1, metaphase 1, anaphase 1, telophase 1. Then we have meiosis 2, no interphase. So from telophase 1 directly to prophase 2, metaphase 2, anaphase 2, and telophase 2. Now, sources of genetic variation. These include crossing over. Now, we talked about crossing over already. This is where when they form the tetrad during synapsis, they can kind of tangle, and then they can break and reform, moving pieces from one to the other. Now, if you look here in our diagram, you'll see that I start with just blue and just red, but due to crossing over, all 4 of these are different at the end. I have one blue, one red, one with red ends but blue in the middle, and one with red in the middle but blue ends. This is just one way to show for that one chromosome. There are different ways it could cross over. Now we have 23 pairs, so we can have this happen on almost all of those. Next, we have independent assortment. And what this means is that during metaphase, when they line up in their pairs, so you have one from mom and one from dad, they're not all lined up the same. So it's not always moms and dads, mom, then dad. It could be mom, dad, dad, mom, dad, dad, or no, dad, mom. There we go. But they can be different ways of lining up, giving us different end combinations. Okay, so our chromosomes, how they line up, how the pair is, whether it's mom or dad first, okay, that has nothing to do with any of the others. So they're independent. And our last source of genetic variation is random fertilization. Random fertilization means that any egg meets with any sperm, and it's random. So an egg or a sperm has 8 million different ways it can be made. That's for humans. So one egg has 8 million ways that it could be made. One sperm has 8 million ways it could be made. And when they combine together, you are a 1 in 64 billion chance of you being you with all your DNA just like it is. So technically, if your parents had 64 trillion other kids, one of them might look just like you. Now that doesn't count for identical twins or triplets. Now, meiosis and mitosis, if we compare them, they are both divisions of the nucleus. Okay, cytokinesis divides the rest of it. In mitosis, we have only somatic cells involved. It's one division creating two diploid daughter cells. And these are clones. They're identical to the original cell, unless there might be some mutation going on. This is what happens from the time you become a zygote until you die, except in special tissue, which would be like the ovaries or the testes. The whole purpose for mitosis is growth and repair. Okay, and none of that special stuff happens during mitosis. During meiosis, that's only to make gametes. So those are our eggs and our sperm. We have to divide cells twice to make eggs or sperm. We wind up with 4 haploid daughter cells. And they're genetically different from the others and from the original. Now, females before birth, follicles are already formed. So kind of like a pre-egg. And then they mature after puberty, about 1 a month. That's average. Sometimes there's 2. Okay? Now, the whole purpose of this is reproduction. No other purpose to it. So here's our visual to show you how they are different. From mitosis and meiosis, we start with, in this case, 2n equals 6, 2n equals 6. And when we're done dividing, in mitosis, we still have 2n equaling 6 for mitosis. But in meiosis, we wind up with cells that are n equals 3. And each one of them are different. All right, make sure to send me a message on Google Classroom, to send me an email, or to see me during office hours if you have questions on this. And we'll be doing more soon.